Well, it's been a great day, and we've heard a lot about trials and drugs and biomarkers and endpoints, but it only matters if we can get the drugs approved so that they can get out to more people and help people in the community and throughout the country. The world looks at what happens here in the U.S. So we're really fortunate that we have a session now to look at the unique issues that are involved in combining immune therapies and how do you label and how would the label work? How do you deal with a drug that might not have activity? And as we've heard about, so many different things that we'll be discussing at the panel. So what we're going to do is we're going to have 4 speakers. We'll have 2 overview talks and then 2 industry perspectives. So it's my pleasure to invite Amy Mckee now from the Food and Drug Administration to talk first. Overview of regulatory and labeling challenges with developing immune checkpoint blockade combination therapies. Amy.
Good afternoon. So what I'd like to do is give you a flavor of what we've been discussing internally at the agency about how to deal with when we get combination trials and applications for all the elements of that regimen for labeled indications, and our current thinking as it is now, which will hopefully be coming forth as a guidance in the near future. So as we've discussed, drug development oncology is moving into combinations with IO products to improve outcomes for patients. One key element to this for the agency is that we require evidence of contribution of effect for each agent in the regimen for which a labeled indication is sought. And so the real bottom line question for everyone is what level of evidence will be acceptable to FDA for the different components of a new regimen to get an indication. And so I just want to start with some definitions that for terminology I'll be using throughout this talk. So we've been calling this cross-labeling in the agency. And just to make clear what I mean by cross-labeling, this is an inclusion of information in product labeling of 2 or more oncology products approved in a combination regimen for a specific indication. Now, this is not co-packaged or co-formulated. This is 2 separate products that are given together in a regimen. And what I mean by regimen is 2 or more therapeutic products that are marketed separately but are approved for use in combination based upon 1 or more adequate and well-controlled trials. So this is the issue. Cross-labeling is actually already here at the agency. We've had over a dozen NDA and BLA applications in the Office of Hematology and Oncology for cross-labeling at this time. The applications have ranged from 2 novel products to 2 supplemental indications and basically everything in between that you can think of. One key component of this may be that supplemental BLAs and NDAs no longer require a user fee under the latest negotiations with industry that have been in effect for a little over a year now. So this may increase our supplemental indications in particular for combinations. So why is this unique to oncology? So the overwhelming majority of clinical trials in oncology measure response rate, either as a key outcome measure or to direct clinical care or both. And this has been going on for decades since the beginning of clinical trials in oncology. So we have a very good history of how agents behave in different disease settings. Also, malignancies generally don't regress spontaneously, so responses attributed to responses can be attributed to treatment intervention and not to the natural history of disease. So we can really rely on response rate as evidence of activity for treatment. Response rate has actually had been a frequent basis of approval in the 1970s before we moved to other outcome measures, but more recently we've been using it for both accelerated and some special situations for regular approval at the agency. The consistency of assessing response rates in oncology trials enables us to estimate the treatment effect contributions for each component in the regimen in oncology compared to other disease areas where they don't have such a surrogate of activity. And just to note, if a company is developing a novel regimen, development and cross-labeling should follow the guidelines that we've set forth in a prior guidance for co-development, which can be found at that website. So I want to show a couple examples of cross-labeling where we've already approved indications in multiple labels so that you can understand what our thinking was in these recent applications. So my first example is dabrafenib and trametinib. Dabrafenib is a BRAF inhibitor that's been frequently combined with trametinib so that any feedback mechanisms for resistance can be overcome. We've approved it as a regimen for melanoma in both the adjuvant and metastatic setting, in non-small cell lung cancer, and most recently in anaplastic thyroid cancer. The initial indication in metastatic melanoma had randomized trials of the combination versus just dabrafenib and the combination versus vemurafenib. For both of those trials, the primary endpoint was overall survival. However, response rate was assessed in both. Dabrafenib individually was already approved in this study as a monotherapy based on a randomized trial that had both PFS and ORR as outcome measures. The subsequent indications had a variety of data in non-small cell lung cancer and thyroid cancer, from randomized trials to single-arm trials. So how did we decide to label both products with these indications? Well, first of all, we knew what dabrafenib monotherapy did in the same or similar settings. So when we added trametinib, we had some idea of what dabrafenib already did in that setting. The biology for this mutation, BRAF, and targeted therapy has been, the responses have been similar across many diseases. I think the one exception so far has been colorectal cancer, where this combination has not been as successful. But in the other three diseases that I've talked about, the responses have been overwhelming compared to what's available. And so with each new approval, we've had evidence from other disease areas, which has given us confidence that both of these products together contributed to the overall effect. And so we've labeled both products with all of these indications. So my second example is pemetrexed with the chemo combination. So pemetrexed was approved for first-line metastatic non-squamous non-small cell lung cancer with pembro and carboplatin as an accelerated approval. Now, pembro also got an accelerated approval in this indication. Thus, we decided that pemetrexed should have the same accelerated approval with the same post-marketing requirement. This approval was initially based on overall response rate with PFS as a secondary endpoint. And the post-marketing requirement is to provide the overall survival results from a randomized trial in the same setting. And again, both the pembrolizumab and the pemetrexed applications have this as a post-marketing requirement. Now, pemetrexed already had multiple approvals in non-small cell lung cancer, and literature up the wazoo showing what sort of effect that you could expect with this product regarding response rates, both as monotherapy and in combination with platinum chemotherapy. So we already knew, we already had evidence for the effect of pemetrexed in these settings. In addition, we had evidence of monotherapy effect from pembrolizumab in this setting. So we knew that pembrolizumab alone could not produce the same effect that pembrolizumab did with pemetrexed and carboplatin in combination. So we had evidence of effect from both pembrolizumab and pemetrexed in this setting. And then finally, we felt inside the agency that it would be difficult and perhaps unethical to conduct a multi-arm trial in which pemetrexed's effect was isolated as monotherapy in relation to the 3-drug combination regimen, given pemetrexed's position as a standard of care in this setting. So for all these reasons, we also gave an indication for pemetrexed. And my final example is fulvestrant, and this has been cross-labeled with both palbociclib and abemaciclib after progression on endocrine therapy in HER2-negative, hormone receptor-positive advanced breast cancer. These are based on an overall response rate with PFS as a secondary endpoint. And the post-marketing requirement is to provide overall survival results from a randomized trial in the same setting. Now, fulvestrant had multiple approvals in advanced breast cancer already as a monotherapy in first-line hormone receptor-positive, HER2-negative, as monotherapy after endocrine therapy in hormone receptor-positive. And we also knew the activity of both palbociclib and abemaciclib as monotherapies in this setting. We know that palbo had very little activity as a monotherapy. Abemaciclib's was a little bit higher, but still, we knew that neither of those agents as monotherapies were able to demonstrate the same effect that the combination regimen had. So we had evidence of activity of fulvestrant as a monotherapy, evidence of what the activity was for abemaciclib and palbociclib as monotherapies in these settings. And so we felt confident that the combination was an improved benefit over any of those monotherapies alone. And finally, again, as in the last setting, we felt it would be difficult or unethical to conduct a multi-arm trial in which the fulvestrant effect was isolated in relation to the combinations given its position as a standard of care. So this is part of an overall goal that we have been interested in the last year or so in terms of making sure that our labels are updated and appropriate for physicians to use. So this is really a multi-pronged approach that we're taking to this. In particular, we are also interested in updating generics labels. A project that we've been working on has identified over 40 oncology products with generics on the market where the products are commonly in use, they're on guidelines, they're sometimes in curative settings. Over half of these aren't even in the PLL formatting that we've been requiring since 2006 because there have been no updates to the label to trigger the PLR labeling. So we feel that these labels really need to be updated, not just potentially with indications, but safety, dosing, the entire label needs to be updated. So we have a project, we're actually nicknaming it Project Renewal, to update some of these labels, and a lot of people in this room have been working on this. And then this is the other half of it. So we're looking backwards to try to update the generics, but so that we don't have to continually update generic labels, we'd like to do cross-labeling to make sure that labels stay up to date with the most current information of how they're actually used in practice and what the clinical trials have shown. So in this way, we are encouraging these cross-labeling supplements. Again, there are no user fees associated with it, so it shouldn't be an added expense to the companies. And the generic labeling product, between these two efforts, we're hoping that the agency will be able to continue to produce labels that are updated and useful to physicians and to the community. So in conclusion, when we think about cross-labeling, we have to think about what position the product is in terms of its current regulatory status to be considered for a combination regimen labeling. Is it approved or an NME? Is it approved in a specific disease or setting? What is all the available data that we have about a particular product so that we can include it in the evaluation of a cross-labeling application? Is it a standard of care product in the community, even if it's not labeled as such? Is it new in that disease context, whereby we would probably require more information about that product? Is there a biologic rationale for why we should cross-label this, maybe across disease settings, the way I mentioned in dabrafenib and trametinib? And finally, we are encouraging companies to submit these cross-labeling applications to us. And again, hopefully we'll have guidance coming out on this shortly. Thank you.